Welcome to this lecture. In our today's lecture, we want to look at a herbarium. When you hear the word herbarium, what comes to your mind? Initially, this term used to refer to a book about medicinal plants because it's from the word herb. Today, the word herbarium refers to a collection of pressed, dried, preserved plant specimens that have been arranged for reference. On our right hand side here, we have a typical herbarium specimen that is for the Tithonia diversifolia species. Now, usually a herbarium specimen can either be pressed and mounted onto a flat sheet, or it can be preserved in a vial with a preservation fluid. Usually in the vials we keep plants that are succulents because it's not easy to press them. These plants in the vials are what we refer to as morphological collections. Now, the plural of herbarium is herbaria. Usually, a herbaria or herbarium has an associated botanical garden. So that now the herbarium has the dried preserved plant specimens, while the botanical garden houses the living collections. So any contemporary herbarium will be having an associated botanical garden. Now, if you have a good quality herbarium specimen, it usually has several uses. So the first use of a good quality herbarium specimen is that it records the presence of a given specimen in a given location. So you'll know that a given species is found in location X. Like for example, if you collected Montia huitei from Kakamega town or Kakamega forest for that matter, you can say this particular species, because of this specimen indicating that collection place is Kakamega, is found in Kakamega. The second use of a good quality herbarium specimen is that the specimen records the appearance of the plant at the time of collection. You know, this appearance can be used to compare with other plants at the same time of collection or at different times of the year. If you collected a plant in March, you can go back to the field in, say, September or November and look at how the plant compares at that particular time or you visited the field. Thirdly, the use of a good quality herbarium specimen is that, you know, when you have your specimen, you can study it at any given time. But for you to study a plant that is found in Mount Elgon, then it requires you to visit that particular place, and you don't just visit at any time, you must visit during the growing season of the plant, otherwise you might not find even that plant. But if you have a good quality herbarium specimen, you can study it any time you want to study it. The fourth use is that a preserved specimen can provide a wide range of information depending on what you wrote on the label. And this information can be useful for historical and phytogeographic studies, as well as monographic and revision research. The fifth use of a good quality herbarium specimen is that, you know, when you have your specimen, you can loan it out just like a library book. So the specimen can be borrowed and loaned and returned at any given time. If you visited the National Museums of Kenya, we have the Department of Botany, and within the Department of Botany the herbarium, the East African herbarium, is housed within that department. So you can borrow a specimen and study it and then return it. The way you will do what you will do when you visit any given library. Previously, we have talked of a nomenclatural type or a type specimen. You see, the type specimen, a given species documents the name of a given taxon, and this is also applicable to a herbarium specimen. A herbarium specimen is a permanent document of the name given to a taxon. So those are the uses of herbarium specimen, and that's why it now becomes very important for you to prepare a good quality herbarium specimen. Now, when you want to collect a herbarium specimen, there are some materials or equipments that you will require. So you are going to look at the equipment and the use. So you can have a plant press or a field press. These are normally used synonymously in some books. These are for pressing the specimen. You will require an old newspaper which will act as the blotter, again for pressing the specimen. You require a field notebook for recording the field information or field data. You will also require various digging tools like the trowel, a large screwdriver, a dandelion digger, a geology pick, and these are for extracting the underground parts and collection of soil, maybe soil samples. You also require a clipper or a knife for collecting woody material. You also require a calico bag. We used to have plastic bags, nowadays we use calico bags for collecting and holding the specimens. You'll require a flexostat for holding plant parts together at bends. We shall be looking at what a flexostat is in the next slide. You'll also require a magnifying lens, of course, for observing plant micro characters. You'll require an insect repellent if you went to collect a plant infested by biting bugs. Then you require an insect repellent to drive off the insects before you collect the plant. You also require small envelopes or small paper envelopes like the ones used for dispensing drugs in chemists, for holding seeds and even fruits of the given plant. With us here is a typical plant press. You can see there's this wooden portion. The upper and the lower portions are wooden. We are having a strap. Then within the plant press we have a ventilator. After the ventilator we have a blotter. After the blotter you have a specimen, then the blotter, then the ventilator. And the same arrangement repeats or recurs. So this is how a plants are arranged within the plant press. You'll also require vials with preserving fluids, and we are saying that these vials are for collecting or holding anatomical, cytological, and even morphological specimens. And we had already said earlier on that they will use the vials for collecting succulent specimens. You will require boots when you are collecting in wet habitats. You will also require darkroom tongs in the event that you are collecting or handling toxic plants like the stinging nettle, which if you just handle with your bare hands, they'll release hypodermic needles, necessitating your body to produce antihistamines, and the experience might not be very interesting for you. Now, you have looked at the materials that are required for collecting a plant specimen. How do you prepare now a herbarium specimen? Now, when you're preparing a herbarium specimen, what you need to do is you are supposed to choose a plant that appears most typical of those within the population of the species you want to prepare. And this plant is supposed to include the flowers, the fruits, or both if you can. It should also have leaves and some underground parts. But what is key here are the flowers and the leaves. Those two, particularly the flower, is sufficient for identifying the particular specimen. So ensure your plant specimen that you're collecting has the flower with it. But I'm not saying you just collect flowers. Ensure the flowers and the leaves and if possible underground parts are present. if you have a plant specimen that does not have the flower, it becomes difficult for one to identify the plant. So we'll say you have collected a sterile specimen. Your specimen is sterile. Now next, ensure that the plant you are collecting has been reduced. You trim it in size so that it fits onto the size of a plant press, which is normally 30 centimeters by 46 centimeters. And again, it should be smaller than a typical mounting sheet, which is normally 29 by 41.5 centimeters. You should also ensure that if you have a large plant and you want to collect it so that it is representative enough, ensure you make the plant into a bend. You bend it into either a V shape, an N shape, or a W shape for this particular plant specimen to fit onto the standard mounting sheet. Now, you also ensure that if at these bends that you have created, you hold the particular plant parts in place using a flexostat. Now, if you have extra large plants that cannot fit onto one mounting sheet, you can then make them into multiple sheets. Now, if the plant is succulent and bulky, sometimes you can sprinkle onto it table salt to help in drawing off the plant's moisture. Aquatic plants are collected by using a high quality mounting paper which is placed under the submerged floating aquatic plant. Then you raise this particular high quality mounting sheet up slowly, and if the plant is lying onto the mounting sheet, then you bring it out of the water. So this is what you are talking about. This is your plant specimen. You see, we are having a leaf. This is the top part of the leaf. This is the underneath of the leaf. So some portions of the leaf should face up while others should face down, because if you don't turn the leaf at this particular point, once the plant is dry, if you try to turn the leaf will break, and therefore you will not have a good specimen. Now on the right side here, these blue portions are the ones we are referring to as flexostats. They are meant to hold the plant at the bends. But then below the plant, you can also you'll apply glue. So the glue helps the plant to adhere onto the mounting sheet. Now it's best to press the plant specimen as soon as possible within a single sheet of old newspaper. Now you can do this in the field using the press. Now it's normally at this stage that pruning is done, so that you just remain with the exact size that you desire to have. Now you also wash if there are any dirt, like maybe you have collected the roots. The roots will have some soil, so you need to wash at this particular point in time before the drying begins. Now usually the specimen is arranged in a manner that it will appear after it has dried. Remember, I told you that if you don't put some portions of the leaf facing up, others down, it will not be possible for it to turn once dry, because they will just break. Now within the plant press, the arrangement of the specimen is usually you have a ventilator, a blotter, a specimen, blotter, ventilator, blotter, specimen again, blotter, then ventilator, etc., depending on how many species or specimens you have collected. Now within a single sheet of newspaper, just place one particular species. Don't put more than one. Only one species should be put in a given folded paper. Now ensure you write as much identification information as possible outside the newspaper in an easily accessible place for reference once you are back to your working station. Now after the specimens have been pressed, the plant press is closed and pressure is applied by means of tightening of the belts. You remember the plant press I showed you in the previous two slides? Now you can have ropes belts or straps for tightening this particular plant press, because as the plant dries, they release moisture and excess slack appears. Slack means the plant press now will appear loose. So you keep on tightening so that now the pressing, a good press achieves its function when you keep on tightening. Now the plant press can be placed in the warm sunshine or on top of the car as one travels. But the best practice, or if you want to obtain the best results, you'll be expected to place this particular plant press in an artificial dryer. Now an artificial dryer is just some wooden chamber that is supplied with some electric bulb, usually about 40 watts. So now the heat from this bulb is the one that is used for drying this particular specimens. Now how long the specimen takes to dry depends on so many variables. The usual time is just about 24 to 48 hours, those are about 2 to 3 days. But sometimes you'll find some fleshy specimens that might take 3 to 6 days. But sometimes you'll find some delicate parts. Some plants are just herbs that are too delicate. So about 8 to 12 hours will be sufficient for this particular plant specimen to dry. Now, a specimen is not dry if it will still limp when you are picking it, or it is cool and moist to the touch. If you find your plant is still cool and moist, return it to the plant dryer and keep monitoring until it dries properly. But now ensure that your plant does not roast in the plant dryer. Let the plant just dry, and as it dries, the integrity of the plant, let the appearance of the plant be as it was when you collected it in the field. Now when you are in the field, all the field information should be entered in a notebook using either a pencil or even a biro pen. Now ensure you collect as much information as possible. Don't leave some information and think that you'll remember and just document and write while you're back to your working station. Some of this information we keep on forgetting, so write as soon as possible as you document the particular information of a given species. Now after drying the specimens, what you need to do is you are supposed now to mount the specimen onto the mounting sheet using glue. Remember we said that if there are bends, you will use a flexostat. Now after you have dried the specimen, you will be expected to treat it. So you treat it by freezing it for 7 to 48 days. The reason why you freeze the specimen is to kill any eggs of bugs. If these eggs are not killed at this particular or inactivated at this particular point, once the plant has been kept in the herbaria, the building now, these eggs will hatch and they will destroy your otherwise good specimen that you have prepared. Now your specimen will require a label, and this is what you need to have on your label. You need to have a scientific name, you need to have a common name, the location or habitat where you collected the plant, the collector, and this is you, the collection date, the collection number, and who identified this particular plant. Now identification is achieved by comparing this plant or the specimen you have with the one which is known. After that, now you prepare your permanent label, and the recorded information is now transferred to this particular label. Now this label can either be handwritten using Indian ink, or you can type it so that you transfer it there. You can now prepare even a label, a voucher label, to establish the authenticity of this particular plant name. Sometimes after you have prepared your label, an authority comes and either verifies that indeed this is the plant that the way you named it, or he varies the name. He says that this is not the name. The name of this particular plant is say maybe Montia huitei. So now the authority now will prepare another label to vary the name, and that label is what we refer to as an annotation label. Now the new correct name is never placed on the original label, it's placed on the annotation label. And that's what you need to do to prepare an herbarium specimen. Thank you.